The Murchison meteorite that fell in rural Australia in 1969 contained over 14,000 organic compounds, including every nucleobase that DNA and RNA are built from, the raw material of life arriving from space already assembled. Subscribe if you want more of the cosmos. This is White Space. Settle in. It is difficult to accept, even as a working assumption, that in a universe containing 2 trillion galaxies, each holding hundreds of billions of stars, the only world where chemistry crossed the threshold into biology is this one. The probability feels wrong, the silence feels wrong, and yet for all our searching we have found nothing. No signal, no fossil, no unambiguous chemistry pointing to something alive. We remain, as far as we know, alone. But the question of whether life exists elsewhere is inseparable from a deeper question. What do we actually mean by life? When we imagine extraterrestrial organisms, we tend to imagine things that look like us, or at least like things we know. Humanoid figures, creatures with bilateral symmetry, limbs, eyes facing forward. This intuition is understandable. It is also almost certainly wrong. Every living thing on Earth is the product of this planet's specific conditions. Strip away those conditions and you strip away the blueprint, change the gravity, the atmospheric composition, the temperature range, the chemistry of the ocean, and you change everything downstream. Body plans, metabolic strategies, the very molecular architecture of heredity. Life on another world would not simply be life wearing a different costume, it would be a different kind of answer to a different set of questions. Consider gravity first. The pull of a world determines how mass is distributed through a living body, how high an organism can grow, how strong its structural materials need to be. On Earth, gravity sets the upper limit on animal size in a remarkably direct way. The relationship between an organism's linear dimensions and its mass follows what biologists call the square-cube law. If you double the height of an animal, its mass increases by a factor of 8. This is why terrestrial giants like elephants and rhinoceroses have evolved thick, pillar-like legs rather than the slender limbs of a gazelle. It is why the blue whale, the largest animal ever to have lived on Earth, can only support its body in the three-dimensional medium of the ocean. Removed from the water, a blue whale does not drown. It suffocates because the weight of its own body collapses its lung capacity. Gravity wins. On a world with weaker gravity, that constraint would relax. Larger body plans would become structurally viable. Organisms could grow taller relative to their skeletal strength, could move more efficiently, could sustain metabolic structures that would collapse under Earth's surface gravity. On a world with stronger gravity, the reverse would apply. Life would be pressed downward, evolution favoring compact, dense, low-slung body plans with correspondingly robust musculature. The creatures of a high-gravity world would be built like nothing we have seen on Earth. Their bones dense as stone, their muscles thick enough to move against a field that never relents. Gravity also shapes the atmosphere, and the atmosphere shapes life in ways that run just as deep. The concentration of gases available for respiration determines which metabolic strategies are viable. In Earth's Carboniferous period, roughly 350 million years ago, atmospheric oxygen reached concentrations around 35%, significantly higher than today's 21%. The result was dramatic. Insects which breathe through passive diffusion along networks of tubes called tracheae could grow to sizes that would be biologically impossible today. Griffin flies, ancient relatives of dragonflies, had wingspans exceeding 70 centimeters. Millipedes reached lengths of 2.5 meters. Scorpions approached a meter. The oxygen-rich air expanded the ceiling on body size for any organism that relied on passive gas exchange. Because the system could supply enough oxygen to fuel larger tissues. When oxygen levels fell at the end of the Carboniferous, the giants went with them. On a world with different atmospheric chemistry, different biological possibilities open. Organisms that use something other than oxygen as their terminal electron acceptor, the chemical that allows energy to be extracted from food, have already been discovered on Earth. We call them anaerobes, and they thrive in environments that would kill most familiar life. In the deep ocean, in sulfur-rich volcanic vents, in sediments far below the reach of sunlight, organisms have evolved biochemical strategies that bear almost no resemblance to the ones that power our cells. Extend that logic to a world with a fundamentally different atmospheric composition, and the life that evolves there becomes genuinely difficult to conceptualize. Not just strange in appearance, but strange in chemistry, in the basic logic of how it converts energy into structure and reproduction. This brings us to one of the most speculative and fascinating questions in astrobiology. Does life require carbon? Carbon is the foundation of every organism we know. Its chemical versatility, the ease with which it forms stable, complex, branching molecular structures, makes it uniquely suited for the task. Carbon can form single, double, and triple bonds. It links readily with hydrogen, oxygen, nitrogen, phosphorus, and sulfur, which are precisely the atoms most abundant in living systems. It can build chains millions of units long without losing structural coherence. Life, as we know it, is essentially organized carbon. But carbon is not the only element with these properties, even if it holds them most fully. Silicon, which sits directly below carbon in the periodic table, shares some of its bonding characteristics. Silicon-oxygen bonds are among the most stable in chemistry. Silicon forms long-chain molecules, and its compounds are enormously diverse, as evidenced by the vast mineral world built from silicon-oxygen frameworks. Theoretical models of silicon-based biochemistry have been developed, and while silicon faces real obstacles that carbon does not, its bonds are less flexible. It struggles to form the double bonds that give organic chemistry much of its richness, and silicon dioxide is a solid at room temperature rather than a gas. Unlike carbon dioxide, the possibility cannot be simply dismissed. Life built around silicon would be slower, simpler, and architecturally less varied than carbon-based life. But in the right conditions, particularly at very low temperatures where silicon chemistry would be more tractable, it might be viable. What it would look like is genuinely beyond our ability to imagine, given the difficulty of predicting the forms that alien life might take. Astronomers and astrobiologists have focused on a more tractable question: where are the conditions that make carbon-based life possible most likely to exist? The answer centers on what astronomers call the habitable zone, the range of orbital distances around a star within which a rocky planet could maintain liquid water on its surface. Water is not life, but water is, as far as we know, indispensable to life. It is the medium in which biochemical reactions occur, the solvent that allows molecules to meet and interact, the carrier that transports materials across cellular membranes. Without liquid water, the chemistry of life as we understand it simply cannot proceed. The habitable zone concept defines a band around each star based on that star's luminosity. Too close, and the planet's water boils away into a runaway greenhouse effect, as happened to Venus. Too far, and water freezes into a global ice layer too thick to permit the liquid ocean beneath. Though here the picture is complicated, moons like Europa orbiting Jupiter may harbor vast liquid oceans beneath their ice shells, kept warm by tidal flexing rather than solar radiation. Planets that lie within their star's habitable zone and have roughly Earth-like masses have been a major focus of exoplanet research over the past 2 decades. The Kepler Space Telescope, which monitored more than 150,000 stars for the slight dimming caused by a transiting planet, found thousands of candidate worlds. Among them are planets that astronomers describe as Earth-like. rocky bodies orbiting in the habitable zones of their host stars with estimated surface temperatures that might permit liquid water. Planets like Kepler-22b, Kepler-186f, and Kepler-452b occupy this category. All of them are too far away to study in detail with current technology. We can measure their size, estimate their mass, infer something about their orbits. We cannot yet know whether they have atmospheres, whether those atmospheres contain water vapor, or whether anything lives on their surfaces. The uncertainty cuts deeper than just distance. Even if we identify the world with every characteristic we associate with habitability, right size, right orbit, right temperature, liquid water on the surface, we would face a problem we have not yet solved. We do not know how life begins. The origin of life on Earth remains one of the most profound unsolved problems in science. We know that it happened. We can trace the biochemical record back more than 3 billion years to microbial communities that were already sophisticated by any measure, but the steps from chemistry to metabolism to the first self-replicating molecule capable of evolution, those steps remain obscure. If we do not understand how life begins, we cannot calculate how likely it is to begin. And without that probability, the question of how common life is in the universe remains genuinely open. While the search for life beyond Earth has generated enormous scientific interest, the solar system itself has continued to hold surprises closer to home. The story of how we came to understand our own planetary neighborhood is, in many ways, a story about the limits of visibility and the persistence of mathematical inference. For most of human history, the solar system was defined by what the naked eye could see. 5 planets were known to the ancients: Mercury, Venus, Mars, Jupiter, and Saturn. Earth itself was not counted among the wanderers, because from the perspective of an earthbound observer, it didn't wander. It was the ground beneath your feet. The count remained at 5 for millennia. Then, in the late 18th century, a musician turned amateur astronomer named William Herschel was conducting systematic surveys of the night sky with a telescope of his own construction. In March of 1781, he encountered an object that behaved differently from a star. It showed a measurable disk rather than a point of light. He reported it initially as a comet. Further observation revealed a nearly circular orbit far beyond Saturn's. Herschel had discovered Uranus, the first planet found in recorded history. The discovery transformed the solar system from a fixed known structure into something that could still yield surprises, and it set in motion a sequence of events that would eventually lead to one of the most contentious debates in modern planetary science. Within decades of Uranus's discovery, astronomers began noticing that its orbit was not behaving quite as Newton's laws predicted. Something was pulling at it, bending its path in ways that could not be accounted for by the gravitational influence of the known planets. The possibility arose: a hidden world massive enough to perturb Uranus across vast distances was moving through the outer solar system. In the mid-19th century, the French mathematician Urbain Le Verrier worked through the calculation, using nothing but the equations of celestial mechanics and the measured deviations of Uranus from its expected path. He predicted the position of an unknown planet with sufficient accuracy that when Johann Galle turned his telescope to the predicted coordinates at the Berlin Observatory in September 1846, Neptune was found within 1 degree of where Le Verrier said it would be. The discovery was one of the great triumphs of Newtonian physics, a planet found with mathematics before it was found with a lens. But the story did not end there. As the 19th century closed and the 20th began, some astronomers became convinced that Neptune alone could not account for all the irregularities in the outer planet's orbits. A further body, more distant, more massive, might be lurking beyond. The American astronomer Percival Lowell, who had spent years convinced that intelligent life had once flourished on Mars, turned his attention to this hypothetical object, which he called Planet X. He died in 1916 without finding it. The search was eventually taken up by the Lowell Observatory in Flagstaff, Arizona. In 1929, the task was assigned to a young self-taught astronomer named Clyde Tombaugh, who had built his own telescopes as a teenager on a Kansas farm. Tombaugh's method was methodical and exhausting. He photographed the same region of sky on 3 separate occasions, days apart, then compared the images looking for any point of light that had moved relative to the fixed stars. In February 1930, he found one, a faint, slow-moving object beyond Neptune's orbit, Pluto. The discovery made international headlines. Pluto was designated the 9th planet of the solar system and remained so for 76 years. But doubts surfaced almost immediately. Pluto was extraordinarily faint, suggesting it was far smaller and less massive than a planet capable of influencing Neptune's orbit should be. Direct measurement of its mass only became possible after the discovery of its largest moon, Charon, in 1978. The result confirmed the suspicion. Pluto's mass is roughly 0.2% of Earth's, nowhere near enough to account for any detectable gravitational effect on Uranus or Neptune. Whatever Percival Lowell had been looking for, Pluto was not it. In 2005, the situation became considerably more complicated. Astronomers announced the discovery of Eris, a body orbiting even farther from the Sun than Pluto and initially estimated to be slightly larger. For a brief period, Eris was described as the 10th planet. The astronomical community faced a choice: accept Eris as a planet, revisit the definition of what a planet is, or find some other solution. In 2006, at the General Assembly of the International Astronomical Union, delegates voted to establish a new category of solar system object, the dwarf planet, and demote both Pluto and Eris to that classification. The decision was controversial and remains so, particularly among planetary scientists who study Pluto and find the distinction arbitrary. But it opened a conceptual space for a much richer understanding of the outer solar system. Beyond Neptune, in the cold reaches of space that receive barely any sunlight, astronomers have catalogued an expanding population of icy bodies. Sedna, Quaoar, Makemake, Haumea, and scores of others. These trans-Neptunian objects follow orbits that range from nearly circular to extraordinarily elongated, and many of them are inclined significantly relative to the plane in which the major planets orbit. Sedna in particular stands out. Its orbit is one of the most elongated known in the solar system, stretching from about 76 astronomical units at its closest approach to the Sun, already well beyond Neptune's orbit, to nearly 1,000 astronomical units at its farthest. No known force can explain Sedna's orbit without invoking the gravitational influence of an as yet undetected massive object. In 2016, Caltech astronomers Mike Brown and Konstantin Batygin published an analysis that added new urgency to this question. Studying the orbital elements of a group of trans-Neptunian objects with extreme orbits, they noticed something striking. The orbits were not randomly distributed. Their computer simulations showed that the observed clustering could be explained by the gravitational influence of an unseen planet, one with roughly 10 times Earth's mass, orbiting far beyond Neptune, on a highly elongated, inclined orbit of its own. The predicted properties of this hypothetical body are extraordinary. Its closest approach to the Sun would be around 200 astronomical units. Its farthest point might be 1,000 astronomical units or more. A single orbit would take approximately 10,000 to 20,000 Earth years to complete. At such distances, it would be extraordinarily faint. Even with modern telescopes, finding it would require surveying enormous regions of sky. Searches are ongoing. The Subaru telescope on Mauna Kea in Hawaii, with its wide field of view and 8-meter primary mirror, is among the instruments engaged in the hunt. An alternative hypothesis proposed in 2019 suggested that the gravitational anomalies could be explained not by a planet, but by a primordial black hole, one formed in the first moments after the Big Bang, when density fluctuations in the early universe may have collapsed small regions of space into compact, intensely massive objects. A primordial black hole of the right mass would be only about 5 centimeters across, invisible to any telescope. The hypothesis attracted significant attention and was subsequently challenged on statistical grounds. The planet hypothesis remains the most widely discussed explanation. What we know for certain is that the outer solar system is stranger, richer, and less fully mapped than we once believed. There may be a world out there, 10 times Earth's mass, tracing an immense ellipse through the dark. Or there may not be. The answer will come from continued observation, and when it does, it will either confirm that our solar system's major architecture is complete, or reveal that there is one more world to add to the family. While the outer solar system remains mysterious, the last 2 decades have transformed our understanding of what orbits other stars entirely. Before 1995, exoplanets were theoretical. We had good reason to expect they existed. The processes that formed our own solar system should operate wherever stars form. But we had no confirmed detections. Then Michel Mayor and Didier Queloz announced the discovery of 51 Pegasi b, a planet orbiting a Sun-like star with a period of just 4 days. It was roughly half the mass of Jupiter, compressed into an orbit tighter than Mercury's. Nothing in our theoretical models had predicted such a thing was possible. Planets, the models said, should form in the cold outer regions of their protoplanetary disks, where volatile ices provide the raw material for giant planets to accumulate. No giant planet should exist this close to its star. And yet, there it was. We now understand that many giant planets migrate inward during formation, carried by interactions with the gas disk in which they form. The disk exerts a braking force on the planet, causing it to spiral toward the star over time scales of millions of years. Some giants stop far from their stars. Others continue inward until the disk dissipates or they reach orbital resonances that arrest their motion. The extreme cases, massive planets orbiting within a few million kilometers of their host stars, have become known as hot Jupiters, and they represent a surprisingly large fraction of the known exoplanet population, partly because they are the easiest to detect. Their short orbital periods produce rapid, repeated transits across the stellar disk. Their gravitational tug on their host stars is strong enough to produce measurable Doppler shifts in stellar spectral lines. Hot Jupiter surface temperatures commonly reach 1,500 degrees Celsius and can exceed 2,500 degrees. At such temperatures, metals vaporize. Iron, chromium, magnesium, sodium, and potassium can exist as atmospheric gases. The detection of these metals has become one of the primary scientific achievements of exoplanet atmospheric spectroscopy. When a hot Jupiter passes in front of its star during a transit, some of the stellar light passes through the planet's atmosphere before reaching our telescopes. Different atoms and molecules absorb light at characteristic wavelengths, leaving distinctive fingerprints in the spectrum. By analyzing which wavelengths are diminished during transit, astronomers can identify what the atmosphere contains. One particularly remarkable example is the exoplanet Mascara 2b, also known by its alternative designation Keld 20b. It orbits a hot blue star in the constellation Cygnus with a period of just over 3.5 days, close enough to its star that its dayside temperature exceeds 2,500 degrees Celsius. Spectroscopic observations using high-resolution instruments have detected the presence of ionized calcium, iron, and sodium in its atmosphere, along with neutral atomic species that exist only because the atmosphere is perpetually blasted by intense radiation. What makes this planet particularly interesting is evidence for strong global circulation in its upper atmosphere. High-altitude winds apparently transport material from one hemisphere to the other, creating a dynamic, churning system rather than a static layered structure. For decades, theorists predicted that hot Jupiters would show signatures of sodium and potassium in their atmospheres, based on models of how atmospheric chemistry should evolve at extreme temperatures. Mascara 2b is one of the clearest confirmations of those predictions to date. The scientific value of studying these extreme worlds extends beyond curiosity. The techniques developed to detect atmospheric chemistry in hot Jupiters, transit spectroscopy, high-resolution cross-correlation spectroscopy, thermal emission measurements, are the same techniques that will eventually be applied to smaller, cooler, potentially habitable worlds. Every detection of an atmospheric fingerprint on a distant planet is a proof of concept for the search that matters most, the search for biosignatures, chemical signatures that can only be explained by the presence of living organisms. Among the most compelling potential biosignatures is photosynthesis, or more precisely the spectral signature of the pigments that photosynthetic organisms use to harvest light. On Earth, forests cover roughly a third of the continental land surface. From space, the presence of vegetation is detectable not just by the naked eye, but by the distinctive spectral signature of chlorophyll. Chlorophyll absorbs light strongly in the blue, violet, and red regions of the visible spectrum, but reflects green light, which is why most plants appear green. This reflection creates what ecologists and remote sensing scientists call the vegetation red edge, a sharp increase in reflectivity at wavelengths just beyond the visible red, in the near infrared. That is a reliable indicator of photosynthetic plant matter. If astronomers could measure the spectrum of sunlight reflected from Earth against the backdrop of the Sun, they would be able to detect chlorophyll from across the solar system. We have actually done this by measuring the Earthshine reflected onto the darkened portion of the Moon. Astronomers have extracted the spectral signature of terrestrial vegetation from afar. It works. The question is whether a similar approach could be applied to exoplanets, and whether the biosignatures we would find there would look anything like chlorophyll. The answer depends on the nature of the host star. Chlorophyll evolved under the specific spectral distribution of solar radiation. Our sun's output peaks in the green-yellow portion of the visible spectrum, which creates an interesting evolutionary puzzle. If the most abundant wavelength of light is green, why do plants reflect it rather than absorbing it? The answer appears to lie in the physics of photosynthetic reaction centers. These molecular machines, where the actual conversion of light energy into chemical energy occurs, can only use photons of a specific energy, essentially the energy carried by red light. Blue photons carry too much energy. When they are absorbed by antenna pigments and the energy is passed toward the reaction center, it must be dissipated as heat along the way to avoid damaging the molecular machinery. Green photons fall in an awkward middle range that most plant pigments cannot efficiently funnel to the reaction centers. The result is that green light becomes evolutionarily something close to waste radiation. It is reflected rather than absorbed because the molecular architecture of photosynthesis on Earth has not found a good way to use it. On planets orbiting different stars, photosynthetic organisms would face different spectral environments and would evolve accordingly. Around a cool red M-dwarf star, the most common type in the galaxy, the available light would be shifted significantly toward infrared wavelengths. Organisms on such a planet, if they developed photosynthesis, would need to harvest whatever light is available. Since the star provides little visible radiation and more near-infrared photosynthetic pigments might evolve to absorb across the entire visible spectrum, reflecting almost nothing. A forest on such a planet might appear deep red, gray, or nearly black because organisms maximizing the absorption of the dim available light would keep almost none of it to reflect. The same logic that makes dark clothing warm in sunlight would make dark pigments advantageous in the perpetual twilight of a dim stellar environment. Around a hot blue A-type star, the situation reverses. These stars flood their planets with intense radiation, particularly in the ultraviolet. Photosynthetic organisms might evolve to reflect the overwhelming portion of light they cannot safely use, potentially appearing white, reflective, or adopting multi-pigment systems in which auxiliary compounds serve primarily a photoprotective function, absorbing and dissipating damaging radiation rather than harvesting it for chemistry. The vegetation on such a world might have a ghostly, pale appearance unlike anything on Earth. In multiple star systems, the challenge becomes more complex. 2 or 3 stars illuminating the same planet would expose its surface to radiation arriving from different directions with different spectra. Photosynthetic organisms would need to optimize against a more complicated light environment, potentially producing dark, broad-spectrum pigments capable of harvesting from whichever star is most accessible at a given moment. All of this is speculative, but it is speculation grounded in understood physical and biochemical principles. And it points to a research program. If astronomers can measure the reflected spectrum of a planet's surface with sufficient precision, they can search for biosignature features that are not chlorophyll specifically, but are consistent with the spectral behavior of a photosynthetic pigment adapted to the planet's stellar environment. The upcoming generation of large ground-based telescopes, instruments with mirrors 30 meters and larger, capable of achieving the contrast ratios needed to pick out planetary light against stellar glare, represent the first realistic opportunity to attempt this. The premise is straightforward. Life, wherever it exists, must harvest energy. If it harvests light, it will leave a spectral imprint. If our instruments are sensitive enough and our models accurate enough, we might detect it. Meanwhile, a very different line of inquiry explores whether life might travel between worlds rather than arising independently on each one. The hypothesis called panspermia holds that microorganisms, or the molecular precursors of life, might survive transport through space on meteorites, in cometary debris, in dust clouds, and seed new worlds upon arrival. The idea is old, proposed independently by several 19th century scientists, and it has generally been treated with skepticism on the grounds that space is a profoundly hostile environment. Vacuum, temperature extremes, intense radiation, and the timescales involved, potentially millions of years of transit, seem to conspire against the survival of anything biological. But biology has repeatedly surprised us about the hardiness of life. An experiment conducted on the exterior of the International Space Station, exposed to open space for extended periods, showed that many types of microorganisms could survive far longer than expected. Bacterial spores, fungal spores, certain types of algae, and even some metazoan eggs demonstrated resilience to conditions that should, by simple reasoning, be lethal. Not everything survived. Fish eggs did not, tomato seeds did not, but approximately 80% of mosquito larvae recovered viable function after exposure. Bacillus spores retained germination capacity. Perhaps the most striking result came from experiments with Deinococcus radiodurans, an organism known primarily for its extraordinary resistance to ionizing radiation, capable of surviving doses that would kill a human being 1,000 times over. The bacterium's resilience comes from multiple redundant copies of its genome, up to 10 copies per cell compared to the 2 copies in a human cell, and from an exceptionally efficient system for repairing radiation-induced DNA damage. In space exposure experiments conducted at the International Space Station, aggregated colonies of Deinococcus survived up to 3 years while suspended outside the station, despite radiation levels roughly 200 times higher than those at Earth's surface. The researchers estimated that if the colonies were thick enough to shield their interior layers from direct radiation exposure, survival across distances of hundreds of light years might be theoretically possible over millions of years, assuming the organisms were embedded in protective rocky material. Separately, samples collected from the external surface of the International Space Station were found to contain marine plankton, microorganisms whose natural habitat is the upper ocean. The explanation proposed was atmospheric transport. Storm systems and ionospheric dynamics can loft material from the ocean surface to extremely high altitudes, from which it might be captured by sampling equipment on orbiting spacecraft. Whether this transport process could continue all the way to orbit, or whether the detected organisms arrived by some other mechanism, remains debated. But the result points to an atmospheric pathway by which biological material might in principle escape planetary gravity wells without any violent event like an impact or a volcanic eruption. The implications for panspermia are significant. If life can hitch rides on ejecta from meteor impacts, survive the journey through space embedded in rocky material, and remain viable for the tens of millions of years that would be required for transfer between neighboring stellar systems, then biology might be a more interconnected phenomenon across the galaxy than we typically assume. Life arising on one world might seed another. A common ancestry might link organisms across dozens of light years, all descended from a single original genesis, wherever that genesis occurred. This possibility raises a profound epistemological problem for the search for life. If we detect biosignatures on a nearby exoplanet, we might conclude that life arose there independently, supporting the hypothesis that life is common and arises wherever conditions permit. But if panspermia is a real phenomenon, the life we detect might share our own ancestry, transported from Earth. Or we might share ancestry with it, transported from elsewhere. The detection of life would not automatically tell us how many independent origins of life have occurred. It might simply tell us that whatever single origin produced our own lineage was productive enough to colonize the neighborhood. The solar system occasionally offers direct evidence of material traveling between stars. In October 2017, the Pan-STARRS telescope in Hawaii detected an object moving through the inner solar system on a trajectory that was unmistakably hyperbolic. Not a closed orbit around the Sun, but an open path that could only be explained if the object had entered from interstellar space and would continue outward into interstellar space after passing through. It was the first interstellar object ever identified within the solar system. It was given the Hawaiian name Umuam'o, meaning roughly a messenger from afar arriving first. Umuam'o arrived at roughly 26 kilometers per second relative to the Sun. As it fell inward, solar gravity accelerated it. At its closest approach, it reached nearly 88 kilometers per second, passing inside Mercury's orbit before swinging back outward. By the time it was identified, it had already passed perihelion and was on its way out. Astronomers had roughly 3 weeks to study it before it faded below detectability. What they found was deeply puzzling. Oumuamua showed no signs of the coma, the diffuse halo of gas and dust that characterizes comets when they approach the Sun. This suggested it was not a conventional icy comet, but it also showed an anomalous acceleration as it moved away from the Sun. It was not slowing at the rate that pure gravitational deceleration would predict. Something was pushing it in active comets. This non-gravitational acceleration is explained by outgassing. Jets of material vaporized by solar heating escape from the nucleus and act as a rocket. But no such outgassing was detected from Oumuamua by any telescope. The object's shape, inferred from the way its brightness varied as it rotated, was highly unusual. One analysis suggested it was elongated like a cigar, perhaps 10 times longer than it was wide. Another analysis found that a flattened, disc-like or pancake-like shape fit the observations equally well. Neither shape has a close analog among the comets and asteroids of our own solar system. Various explanations have been proposed. One hypothesis, proposed by a group including astronomers Douglas Hamilton and Stephen Desch, suggested that Oumuamua might be composed primarily of solid nitrogen, the same material that makes up much of Pluto's surface. Solid nitrogen is highly reflective, which would explain why the object appeared so bright relative to its estimated size. And as solid nitrogen is heated by sunlight, it sublimates directly from solid to gas without forming a liquid phase. This sublimation could produce the observed non-gravitational acceleration without producing a visible coma or detectable outgassing, because nitrogen is transparent at visible wavelengths. Under this model, Oumuamua would be a fragment chipped from the frozen nitrogen mantle of a Pluto-like world orbiting another star, a chip of another solar system's outer margin, flung outward by an ancient collision and drifting for tens of millions of years before passing through ours. The origin question is no less interesting. Tracing Oumuamua's path backward, astronomers attempted to identify where it came from. Its trajectory pointed toward the constellation Lyra, and the star Vega lies roughly in that direction. But Vega was not near that position of the sky 600,000 years ago, when Oumuamua would have passed through the region if it were traveling from there. Current best estimates suggest the object was ejected from a star system in the constellation Keel approximately 45 million years ago, from a system that has since shifted in position due to the precession of the celestial sphere. The passage of Oumuamua through the solar system lasted only weeks from a human perspective, but its scientific legacy has been enormous. It demonstrated that interstellar objects pass through the solar system and that they do so, presumably, regularly. We found Oumuamua only because we happened to be looking in the right direction at the right time, with telescopes sufficiently sensitive to detect it. Statistical models suggest that objects of this size pass through the inner solar system several times per year. We simply lack the continuous, deep, wide-field surveys needed to catch most of them. The second interstellar object, Comet Borisov, was discovered less than 2 years later, in August 2019. And unlike Oumuamua, it behaved in every respect like a standard cometary body, releasing gas and dust as it approached the Sun, displaying a clear coma and tail. The contrast between the two objects suggests that interstellar material is diverse, just as solar system material is diverse. As we search for life across cosmic distances, it is worth pausing to consider how tenaciously it clings to existence in the environments we already know. Earth's biosphere extends into conditions that would have seemed obviously incompatible with life to any 19th century naturalist. The lesson these organisms teach is that life's envelope of tolerance is far wider than we once assumed. The deep ocean, below the reach of sunlight and under pressures that would crush unprotected human tissue, hosts thriving ecosystems built not on photosynthesis but on chemosynthesis, the extraction of energy from chemical reactions involving hydrogen sulfide, methane, and other volcanic compounds. At hydrothermal vents, where superheated water at temperatures exceeding 300 degrees Celsius jets upward from the seafloor, communities of tube worms, shrimp, crabs, fish, and microbial mats persist. Some of these organisms tolerate temperatures that would denature the proteins of most known life. The Pompeii worm, Alvinella pompeiana, lives in tubes attached to hydrothermal vent chimneys and tolerates sustained temperatures of 50 degrees Celsius, while its tail end extends into water approaching 80 degrees. It manages this gradient by maintaining a layer of bacteria across its back that may provide thermal insulation or supplementary nutrition. Nothing about its existence would have seemed biologically plausible before it was found. High in glaciers, at altitudes where most organisms cannot survive, a group of wingless insects called ice crawlers or grilloblattids have made their home in conditions that would kill most other insects outright. These ancient lineages, close relatives of crickets and grasshoppers, are most comfortable at temperatures near 4 degrees Celsius. Heat is lethal to them. Temperatures above 15 degrees cause their nervous systems to malfunction. They live their entire lives in a world of ice and rock, feeding on organic material blown up by wind from lower elevations, mating and laying eggs in frozen soil, and completing larval development over periods of up to 7 years. A single adult may live for 20 years in an environment where most organisms could not survive a single night. In the African savanna, a very different extreme survivor has become something of a celebrity among biologists studying aging and cancer. The naked mole rat, Heterocephalus glaber, is a small rodent that lives in underground tunnel systems in East Africa, spending its entire life below ground in colonies of several dozen individuals ruled by a single breeding queen. It is almost completely blind and functionally hairless. It lives in an atmosphere with carbon dioxide concentrations far higher than most mammals can tolerate, and oxygen concentrations far lower. In conditions of severe hypoxia, oxygen deprivation, a naked mole rat can sustain itself for up to 30 minutes with essentially no oxygen intake by switching its cellular metabolism from glucose to fructose, using a metabolic pathway more commonly found in plants. A human brain begins to suffer irreversible damage after 3 minutes of oxygen deprivation. A naked mole rat's brain, at the same level of deprivation, continues to function for half an hour. Its maximum lifespan approaches 30 years, roughly 10 times longer than would be predicted for a rodent of its size. It does not develop cancer in laboratory conditions. The mechanisms underlying its longevity are actively studied as potential models for human medicine. The organisms that most capture the imagination of those thinking about life on other worlds are the extremophilic microbes, bacteria and archaea that have colonized environments defined by conditions once considered universally lethal. The Inococcus radiodurans, already mentioned in the context of panspermia experiments, can withstand radiation doses of 25,000 Gray, a dose that would kill a human being at 1/10 of 1% of that level. The bacterium's genome is replicated in multiple copies and backed by molecular repair systems that can reconstruct a completely shattered chromosome within hours. Thermophilic archaea grow optimally at temperatures exceeding 100 degrees Celsius, living in geysers and volcanic hot springs where water remains liquid only under pressure. Halophiles thrive in saturated brine, where most organisms would lose water by osmosis and die within minutes. Psychrophiles live in Antarctic ice, growing slowly in thin films of liquid water around ice crystals, at temperatures down to -20 degrees Celsius. These organisms collectively expand the definition of the habitable zone to include not just the temperate surface zone around stars, but any environment where liquid water, some energy source, and the necessary chemical elements coincide. By that expanded definition, potential habitats in our solar system become considerably more numerous. The subsurface oceans of Europa and Enceladus, the methane lakes of Titan, the deep subsurface of Mars, where ancient liquid water may still persist in geothermal pockets, the aerosol clouds of Venus, where temperatures and pressures are actually quite moderate despite the hellish surface conditions below. The nearest of all potentially habitable worlds lies around the nearest star. Proxima Centauri is a red dwarf located 4.24 light-years from the Sun, the closest stellar neighbor we have. It is a faint, cool star, only about 12% of the Sun's mass, barely distinguishable from a large planet at the boundary of hydrogen fusion. It is gravitationally associated with the Alpha Centauri binary system, two Sun-like stars orbiting each other. But it orbits at such an enormous distance from the binary pair that it takes nearly 600,000 years to complete one circuit around them. In 2016, astronomers announced the discovery of an Earth-mass planet orbiting Proxima Centauri within its habitable zone. Designated Proxima Centauri b, it orbits at a distance of roughly 7.5 million kilometers, 20 times closer to its star than Earth is to the Sun, and completes an orbit in just 11.2 days. Despite this intimacy, the planet is not necessarily hot. Proxima Centauri emits only about 0.17% of the Sun's luminosity, and at that distance, the planet receives roughly 65% of the radiation Earth receives from the Sun, potentially consistent with surface temperatures that permit liquid water. Estimates suggest a mean surface temperature somewhere between 7 and 15 degrees Celsius if the planet has an Earth-like atmosphere, which is mild enough for liquid water to exist in abundance. Two additional planets have since been proposed. Proxima Centauri d, orbiting closer than b and discovered through high-precision radial velocity measurements in 2022, has a mass of roughly 1/4 of Earth's and orbits in just 5 days. Too close to be habitable. Proxima Centauri c, if it exists, is a potential super-Earth in a distant cold orbit, likely more reminiscent of an icy Neptune than a warm terrestrial world. All attention from an astrobiological standpoint remains focused on Proxima b. The challenges facing any life on Proxima b are substantial and potentially prohibitive. Proxima Centauri is a flare star. Like most red dwarfs, it is magnetically active, generating stellar eruptions that can be orders of magnitude more powerful in proportion to the star's baseline luminosity than even the most intense solar flares from our own Sun. In 2017, a particularly intense event caused Proxima Centauri's brightness to increase by a factor of 1,000 in approximately 10 seconds. An eruption producing roughly 10 times more energy in the X-ray band than the most powerful solar flares observed from Earth. Such events occurring regularly over billions of years would subject any planet in the habitable zone to intense bombardment by ionizing radiation and energetic particles. The consequences depend critically on whether Proxima b has retained an atmosphere, and whether that atmosphere is backed by a magnetic field capable of deflecting the stream of charged particles that accompanies each flare. Earth's magnetic field, generated by convection in its liquid iron outer core, deflects much of the solar wind and channels energetic particles toward the polar regions rather than the surface at large. Without such a field, even a substantial atmosphere can be gradually eroded by the stellar wind, a process that may explain why Mars, which appears to have lost its global magnetic field billions of years ago, now has an atmosphere less than 1% as thick as Earth's. Whether Proxima b has a magnetic field is unknown. The planet is likely tidally locked to its star, its rotation period synchronized with its orbital period, so that one face perpetually points toward the star, while the other face is eternal darkness. Tidal locking suppresses the differential rotation of the planetary interior that drives magnetic dynamo generation on rapidly rotating worlds like Earth. It might be more similar in this respect to the Moon, which has no global magnetic field, or Mars. The potential absence of a magnetic field would make Proxima b considerably more vulnerable to atmospheric erosion and surface radiation than the raw energy budget calculation would suggest. And yet, some pathways to habitability remain open. If Proxima b formed in the cold outer regions of the protoplanetary disk and migrated inward, as many planets are now thought to have done, it would have arrived at its current orbit with a substantial inventory of volatile materials, water, carbon dioxide, nitrogen, sufficient to maintain an atmosphere against stellar erosion for billions of years. A thick enough atmosphere would provide the physical shielding needed to bring surface radiation down to levels compatible with life. Organisms on such a world would not need to be unusually radiation resistant by the standards of terrestrial extremophiles. They would simply need an atmosphere to hide under. An alternative hypothesis invokes biological response rather than physical shielding. Certain marine organisms on Earth have evolved the ability to convert harmful ultraviolet radiation into lower energy visible light through fluorescence using specialized protein complexes. This conversion serves a photoprotective function, harmlessly dissipating energy that would otherwise damage DNA and cellular machinery. Some researchers have speculated that organisms on a world bathed in intense UV and X-ray radiation might evolve similar or analogous mechanisms, potentially becoming so efficient at this conversion that they glow visibly in wavelengths detectable from space. A planet covered in bioluminescent life responding to the perpetual flare activity of its host star would present a distinctive spectral signature unlike any other natural phenomenon. Whether Proxima b is habitable remains unknown. We cannot yet measure its atmospheric composition. We cannot determine whether it has a magnetic field. We do not know its radius with precision, and therefore do not know its bulk density, which would tell us whether it is rocky or water world-like in composition. What we do know is that it is close enough to potentially study with the next generation of telescopes. Close enough that the James Webb Space Telescope has already attempted to constrain its atmospheric properties, and that future instruments may do much better. An even more direct approach has been proposed. The Breakthrough Starshot initiative, announced in 2016 with backing from Silicon Valley investors and Stephen Hawking among its public advocates, aims to develop a fleet of gram-scale spacecraft, tiny chips of electronics attached to reflective sails 4 meters square that could be accelerated by a directed laser array to roughly 20% of the speed of light. At that velocity, the journey to the Alpha Centauri system would take approximately 25 years. The craft would pass through the system in a matter of hours, capturing images and spectral data before sailing onward into the void. The data would take another 4 years to reach Earth. Total elapsed time from launch to data return. Roughly 30 years, within a human lifetime. The technical challenges are enormous. Building a laser array powerful enough, engineering spacecraft that can survive the interstellar medium for 25 years at that velocity. Developing communication systems capable of transmitting data across 4 light years from a gram-scale transmitter, but none of them appears to violate fundamental physics. The project is in a sustained engineering development phase. Whether life exists on Proxima b, we do not yet know. The universe has arranged things so that our nearest stellar neighbor has a planet in its habitable zone, and that planet is close enough that we might, within the next few decades, have real data about its atmosphere. This is either a profound coincidence or a reflection of the fact that habitable zone planets are common enough that they appear even around the closest star we can examine. Either interpretation is remarkable. We are at the beginning of this investigation. The questions we are asking, whether life exists elsewhere, how common it is, what forms it takes, whether it can travel between worlds, are among the oldest questions humanity has carried. For most of our history, they were philosophical, incapable of being tested. Now, for the first time, we have instruments sensitive enough to search for chemical signatures of life across dozens of light years. We have spacecraft that have escaped the solar system entirely. We have detected thousands of planets orbiting other stars and begun, tentatively, to measure the gases in their atmospheres. We have found organisms on our own world that could in principle survive the journey between worlds. We have watched an object from another stellar system pass through our own solar system and leave before we could reach it. The universe is not making itself easy to read, but it is no longer entirely closed to us. The answers, when they come, will not arrive all at once. They will come incrementally, a spectrum here, a detected molecule there, a climate model constrained by new data, a gravitational wobble that turns out to be caused by something alive. We will not one day flip a switch and know. We will accumulate understanding piece by piece until the picture resolves. What the picture will show, we cannot say, but the act of looking carefully, methodically across every wavelength and every scale is itself the most honest response a mind can make to a universe this large and this old and this incompletely understood. There is something humbling and perhaps instructive in the catalogue of Earth organisms that seem already prepared for conditions we associate with other worlds. To move through this list is to confront the realization that life on our planet has already explored environmental extremes that once seemed absolute barriers and found ways through. Consider the deep-sea cephalopod known by taxonomists as Magna pinna, sometimes called the big fin squid. This creature inhabits the bathypelagic zone at depths between 2,000 and 5,000 meters, where pressure exceeds 200 atmospheres and sunlight has never penetrated. It has 10 arms, like most squid, but its proportions are unlike anything that lives near the surface. The arms extend enormously relative to the body, with reports suggesting total spans of several meters on large individuals. It has been encountered alive only a handful of times in recorded history, almost always by deep-sea remotely operated vehicles that happened to direct their lights into an abyss where the animal was drifting. Nothing is known about its reproduction. Nothing is known about its prey. The limits of its depth tolerance have never been tested, because we have never observed it at its deepest or shallowest habitual range. It simply exists in the dark under pressures that would collapse a steel drum about which we know almost nothing. There is something about this particular ignorance that captures the spirit of the entire field. Life has found ways into spaces we never thought to look, building its presence there over millions of years before we arrived with our lights and our lenses, higher up in environments defined not by pressure, but by radiation. The braconid wasps represent a set of survival adaptations that seem almost purpose-built for survival in extreme environments. These are parasitoid insects, organisms that lay their eggs inside the living bodies of other insects, where the larvae develop while consuming their host from the inside. What distinguishes braconids in the context of radiation resistance is the degree to which their physiology can withstand ionizing damage that would be immediately lethal to vertebrates. Laboratory tests have shown that some braconid species can survive radiation doses of approximately 1,800 gray, a unit that represents the absorption of 1 joule of energy per kilogram of tissue. For context, a dose of 8 to 10 gray is sufficient to kill a human being with near certainty within weeks, through widespread disruption of the rapidly dividing cells of the immune system and gut lining. Braconids withstand doses twice into times larger, partly through cellular mechanisms that efficiently repair double-strand breaks in DNA, and partly through physiological traits associated with insects generally. A distributed nervous system, a decentralized body plan, and the absence of the sensitive, rapidly dividing stem cell populations that make vertebrate tissue so vulnerable to radiation damage. Perhaps most remarkable of all, in terms of what it suggests about the portability of life through hostile environments, is the tardigrade. These microscopic animals, also called water bears or moss piglets, names that somehow fail to capture their significance, are found on every continent in every major habitat, from the highest altitude mountain peaks to the deepest ocean sediments, from tropical rainforests to Antarctic ice sheets. They measure between 0.5 millimeter and 1.5 millimeter as adults. Under favorable conditions, they go about a life recognizable in its broad outlines, eating, reproducing, occasionally falling prey to nematodes and other microscopic predators. What makes them extraordinary is what happens when conditions become unfavorable. When a tardigrade faces environmental stress, desiccation, extreme temperature, radiation, vacuum, chemical toxins, it can enter a state called cryptobiosis. Its body contracts, expelling nearly all its water content. Its metabolism drops to less than 0.01% of its normal rate. Its cellular machinery comes essentially to a halt, and the organism enters a condition that is neither life nor death in any conventional sense, but a kind of suspended potential, alive in chemistry but not in process. In this state, a tardigrade can survive exposure to temperatures from minus 272 degrees Celsius, just above absolute zero, to 150 degrees Celsius. It can survive pressures 6 times greater than the deepest ocean trench. It can survive radiation doses of up to 570,000 gray orders of magnitude, beyond what kills every other multicellular organism we know. In 2007, a sample of tardigrades was brought to low Earth orbit and exposed to open space for 10 days, including direct exposure to unfiltered solar, ultraviolet, and cosmic radiation. When returned to Earth and rehydrated, a significant proportion survived and subsequently reproduced successfully. The tardigrade's resistance to desiccation is explained in part by a protein unique to tardigrades, called damage suppressor or DSUP, which binds to chromatin, the complex of DNA and protein that makes up chromosomes, and physically shields it from the water-derived radicals that cause most radiation damage. The protein appears to have no homologue in any other organism. It was evolved, presumably by organisms living in environments where episodic, complete desiccation was a recurring selective pressure, lichen-covered rocks, dried-up forest litter, desert soils. The radiation resistance appears to be a byproduct of the same molecular toolkit that manages desiccation damage. The existence of the tardigrade tells us something important. Multicellular animal life has evolved the capacity to withstand conditions essentially equivalent to open space. Whether any organism could actually survive the timescales involved in interstellar travel is a separate question, involving millions of years of transit. But the tardigrade demonstrates that the boundary between life and the most hostile conditions in the universe is less sharp than it appears. Against this background of extreme terrestrial life, the question of viruses in the context of astrobiology becomes surprisingly nuanced. Viruses occupy a contested position in the hierarchy of living things. They possess genetic material, either DNA or RNA, enclosed in a protein coat, and they can evolve by natural selection. But they lack the cellular machinery to reproduce independently. A virus outside a host cell is biologically inert, a molecular structure with no metabolism, no active process, no agency of any kind. Contact with a compatible host cell transforms it into something that hijacks the cell's synthetic machinery and produces hundreds of copies of itself. In the interval between contacts between one host and the next, a virus exists as what virologists call a virion, a durable, chemically stable package of genetic information and the proteins needed to deliver it. This distinction matters for questions about cosmic transport. A virion, particularly one encased in a robust protein shell, is a relatively hardy object. Polioviruses and bacteriophages, the viruses that infect bacteria, have been shown to survive vacuum and ionizing radiation conditions in laboratory tests. Some bacterial viruses can remain structurally intact and potentially infectious after exposure to conditions broadly similar to the space environment if they are protected against direct high-energy photon flux by embedding in ice or rocky material. The question of whether virions could survive an interstellar journey is separate from the question of whether they could survive near-Earth space exposure. And the answer is probably not on the timescales that interstellar distances require. But within a single planetary system, or between planets sufficiently close that impact ejecta could make the crossing in thousands rather than millions of years, the possibility of viral transport cannot be entirely dismissed. The more immediate implication for astrobiology is methodological rather than narrative. Variants, as stable molecular structures encoding genetic information, constitute a category of biological marker that could potentially be detected in planetary environments if the right sampling tools and detection systems were available. The standard approach to detecting microbial life involves looking for metabolic activity, chemical disequilibrium, or the morphological signatures of cells in microscopy. Variants, being acellular, would be invisible to most of these approaches. An astrobiology mission that found no evidence of microbial life on a Martian surface sample would not necessarily have ruled out the presence of viral genetic material embedded in the soil or frozen in subsurface ice. The realization has prompted some astrobiologists to argue for including viral detection capabilities in future life detection instruments. Not because viruses without hosts would be alive in any meaningful sense, but because their presence would be strong evidence that hosts exist or recently existed nearby. The origin of viruses on Earth is itself instructive. Current evidence suggests that viruses did not precede cellular life, but arose from it, most likely through multiple independent events in which fragments of cellular genetic material escaped their cells of origin and evolved the ability to re-enter and replicate in new cells. Under this model, viruses are evolved cellular escape artists rather than primitive predecessors of life. If life arose on another world, it would almost certainly generate its own viral satellites, molecular parasites arising from the same cellular machinery that makes any complex life possible. To find evidence of viruses on another world would therefore be, in a sense, to find evidence of complex cellular life already present or recently departed. This brings us full circle to the question that underlies all the science described here, the question of origin. We can describe the conditions that favor life. We can identify the worlds where those conditions might exist. We can study the organisms that push the limits of where life can go. What we cannot yet do is explain why chemistry becomes alive. The transition from a rich prebiotic chemistry, amino acids, nucleotides, lipid membranes, catalytic RNA molecules, to the first organism capable of Darwinian evolution is a transition we cannot recreate in the laboratory, though we are closer to understanding its components than at any previous moment. We know that amino acids form spontaneously in simulated early Earth conditions, and in space they have been found in meteorites, including in the carbonaceous chondrite that fell near Murchison, Australia in 1969, which contained more than 70 different amino acids on analysis. We know that nucleotides, the building blocks of RNA and DNA, can also form abiotically. We know that lipid membranes can self-assemble from simple fatty acids, creating the enclosed compartments that allow chemistry to be concentrated rather than diluted into an open ocean. We know that RNA molecules can catalyze chemical reactions, including reactions involved in their own replication, raising the possibility that life began as a self-replicating RNA system before DNA and proteins took over their respective roles. What we do not know is how likely any of these steps are, individually or in sequence. The question of probability haunts every estimate of how many living worlds there might be if the origin of life is highly probable given the right conditions, if chemistry reliably crosses the threshold into biology whenever it has long enough to run, then the galaxy should be teeming with life. And the absence of any detected signal in our searches so far implies either that life is rare enough on cosmic scales, or that the gulf between microbial life and technological civilization is wider and more rarely crossed than we assume. If the origin of life is extraordinarily improbable, a sequence of events so unlikely that it has happened only once or a handful of times in the observable universe, then our existence is a statistical outlier of the most profound kind, and the silence of the cosmos is simply what silence sounds like when you are alone. We do not know which of these possibilities is closer to the truth. We may not know for decades. The tools that would tell us the atmospheric spectrographs capable of detecting biosignatures on rocky exoplanets, the missions capable of sampling the subsurface oceans of Europa and Enceladus, the future observatories capable of imaging exoplanet surfaces at sufficient resolution to detect the vegetation red edge analog of another world are either under construction, in development, or still on the drawing board. What we have learned in the time since 51 Pegasi B showed us that planetary systems are radically more diverse than our models predicted, is that the universe does not organize itself according to our prior assumptions. The solar system we thought we understood turned out to have a hidden ninth body that we still haven't found. The exoplanet population turned out to be dominated by planet types, hot Jupiters, super-Earths, mini-Neptunes, that simply do not exist in our own system. The interstellar medium turned out to deliver visitors to our solar system. Extremophiles turned out to survive conditions that should, by the standards of 20th century biology, be impossible for life to tolerate. The universe has a consistent record of being stranger than expected. That record does not guarantee that life is common, but it should make us cautious about concluding that it is rare based on the narrow sample of what our own world has shown us. Every scientific principle that governs physics and chemistry applies uniformly across the cosmos. The same elements that make up Earth's oceans are present in the atmospheres of stars 50,000 light years away. The same gravitational laws that shaped our solar system shaped every other. The same basic organic chemistry that preceded life on Earth plays out in interstellar clouds and in the ice mantles of cometary nuclei scattered across the outer solar system. If life is a consequence of chemistry operating under certain conditions for sufficient time, and if those conditions are common enough to have arisen once within a few astronomical units of an unremarkable star in one of the Milky Way's outer spiral arms, then there is reason to think they arise elsewhere too. Perhaps rarely, perhaps not as often as optimists hope. But not only here. The answer is out there, written in the spectra of distant atmospheres, in the chemistry of subsurface oceans we have not yet reached, in the radio silence of a universe that may simply be quieter than a technological civilization would expect, or may be sending signals we have not yet learned to recognize. We are early in this search, far earlier than it sometimes seems, when we review the technologies we have assembled and the distances we have managed to probe. The sky is old. The stars have been there for billions of years. We have been looking systematically and scientifically for a matter of decades. There is time, there are more instruments coming. There are more questions worth asking. And the next signal, the next spectrum, the next unexpected object falling through the solar system on a hyperbolic path might be the one that changes everything. The history of discovery in this field teaches repeatedly that the apparent silence or emptiness of a region often reflects the limits of our instruments rather than the limits of what is actually there. The outer solar system was for most of human history nothing but darkness beyond Saturn. Then Uranus emerged from the noise, then Neptune from a mathematical prediction, then Pluto from a systematic photographic survey, then an entire population of icy bodies extending for tens of thousands of astronomical units in every direction. Each new instrument, each new survey strategy, each new theoretical framework opened a new layer of complexity that was always there, waiting for the right tool to reveal it. The same progression is visible in exoplanet science. Before the first confirmed detection in 1995, we operated on assumptions derived from a sample of exactly one planetary system. Everything we thought we understood about how planets form, how they migrate, how they acquire atmospheres, how they interact with their host stars, all of it was built on the single data point of our solar system. The discovery of hot Jupiters shattered that framework within months. The subsequent discoveries of super-Earths, planets between 1 and 10 times Earth's mass, with no analog in our own system and yet apparently the most common type of planet in the galaxy, shattered it further. Mini-Neptunes, warm Jupiters, circumbinary planets orbiting 2 stars simultaneously, planets with densities so low they would float in water, planets with surfaces hot enough to melt iron. The zoo of possibilities has expanded with every new telescope and every new analysis technique. Against this background of diversity, the statistical argument for the abundance of life becomes more compelling, but also harder to evaluate. If the habitable zone around a star can be occupied by many different kinds of planet, and if many of those planets might support life in some form even if their conditions differ substantially from Earth's, then the frequency of inhabited worlds might be higher than a strict Earth-centric analysis would predict. But if life requires a specific and narrow set of conditions, the right planet size, the right orbital parameters, the right stellar environment, the right chemical starting conditions, then the sheer abundance of planets in the galaxy does not automatically imply an abundance of life. This uncertainty is formalized in what is sometimes called the Fermi paradox, after the physicist Enrico Fermi, who raised the question informally at a 1950 lunch conversation. The argument is simple. If intelligent life is common in the galaxy, and if any technological civilization produces interstellar travel or communication at some point in its development, then given the age of the galaxy, roughly 13 billion years, some civilization should have colonized or at minimum communicated across the entire Milky Way by now. The galaxy is large, but not infinitely so, and the timescales available dwarf any conceivable travel time, and yet we detect nothing. No alien signals in the radio frequencies we have monitored, no megastructures around distant stars that would imply the engineering of civilizations harnessing stellar energy at scale. No visitors in the geologic record. Several resolutions have been proposed ranging from the optimistic to the deeply unsettling. Perhaps intelligent civilizations are common, but prefer communication modes we have not yet thought to monitor: neutrino beams, gamma-ray bursts, quantum channels. Perhaps the distances involved are simply too great for any sustained communication, and each civilization remains effectively isolated by the speed of light. Perhaps the transition from microbial to complex to intelligent life is so improbable that it has happened only a handful of times in the observable universe. Perhaps intelligent civilizations reliably destroy themselves before reaching the stage of interstellar capability through war, resource depletion, technological accident, or some other filter that our own civilization has not yet passed. None of these resolutions is satisfying in the sense of being confirmed. All of them remain speculative. The silence of the cosmos, like so much else in this domain, is data but not yet interpretation. What we can say is that every line of evidence we have gathered points to a universe that is not hostile to the chemical precursors of life, is not structurally hostile to planetary systems, and is not obviously hostile even to the kind of complex molecules that biological systems require. The organic chemistry of space is rich: amino acids, sugars, nucleobases, polycyclic aromatic hydrocarbons. These compounds form spontaneously wherever carbon, hydrogen, oxygen, and nitrogen interact under the energy of starlight or cosmic rays. The Murchison meteorite recovered from rural Victoria, Australia, after a 1969 fall contained more than 14,000 distinct organic compounds on analysis, including all 5 nucleobases used in DNA and RNA, and a large fraction of the 20 amino acids that living systems use in protein synthesis. The interstellar medium, the diffuse gas and dust filling the space between stars, contains detectable concentrations of formaldehyde, methanol, glycolaldehyde, a simple sugar, and even recently traces of propylene oxide, a molecule with the property of chirality, meaning it exists in 2 mirror image forms that are otherwise chemically identical. That last detection is significant. The life we know uses only one chirality of amino acid and one chirality of sugar, a preference whose origin is connected to the deepest questions about how life's molecular machinery got started. None of this proves that life has started elsewhere. It proves that the raw materials are distributed across the galaxy and the universe, that the chemistry of life is not exotic, but rather a natural consequence of the element distribution produced by stars and supernova nucleosynthesis over billions of years. If the same chemistry that assembled the first living molecules on Earth is running on the surfaces and in the atmospheres of trillions of other worlds, the question of life's frequency becomes, at some level, a question about kinetics. How fast does the chemistry move, and how often does it cross the threshold? We are building the instruments to start answering that question. The James Webb Space Telescope has already measured the atmospheric spectra of several exoplanets with an accuracy that was impossible from the ground, detecting carbon dioxide, water vapor, methane, and sulfur dioxide in the atmospheres of worlds across distances of dozens to hundreds of light years. These are not biosignatures. They can all be explained by non-biological chemistry, but they are proof that the technique works at interstellar scales, and that the atmospheric signatures of Earth-like planets are within reach of the next generation of observatories. The extremely large telescope currently under construction in Chile will have a primary mirror 39 meters in diameter, 3 times larger than any existing telescope. When complete, it will be capable of directly imaging the reflected light from rocky planets in the habitable zones of nearby stars, and of measuring the spectra of those reflected signals with sufficient resolution to detect molecular features. If there is a planet within 20 light-years of Earth, whose atmosphere contains oxygen, methane, and nitrous oxide simultaneously, a combination that is thermodynamically unstable and therefore implies a continuous biological source, we will probably find it within the lifetime of people already born. If such a planet does not exist within that distance, it will tell us something about the frequency of life that will force a reckoning with our assumptions about how common biology is in the cosmos. Either outcome would be one of the most profound scientific results in human history. We began this exploration with a simple observation. It is hard to believe that Earth is the only inhabited world. We ended in approximately the same place, not because the science has failed to advance, but because the science has clarified the depth of the question. We know more than we have ever known about the conditions that could support life. We know more about the range of planetary environments in the galaxy. We know more about the hardiness of life on our own world and the fragility of the conditions that support it. We know that the chemistry of life is not confined to Earth, but is woven through the molecular fabric of the universe. And we know that the step from chemistry to biology, from organized matter to living matter, is a step we have taken once that we do not yet fully understand. That incompleteness is not a failure. It is an invitation. The universe has been here for 13.8 billion years. Life on Earth has been here for nearly 4 billion of them. Technological civilization has been here for a few thousand. We have been looking beyond our atmosphere with instruments capable of detecting planetary atmospheres for less than a decade. We are early. We are very early. And the universe, by any measure we can apply, is very large.